Hi everyone, this is Clara, and the purpose of this video is to showcase the dataset of my team's patient group. We decided to work on lung cancer, and we found a really unique one on the Cancer Imaging Archive that shows patients that were diagnosed with 4 different types of lung cancer. So we said, okay, this is perfect. Each one of us, because we are 4 members, can work on one of these types of cancer, and because 5 different radiologists approved of the location of the tumor, we were really confident that we would be able to determine where the tumor is. And not only that, but they provided the annotation files of the bounding boxes, so we can see way better through all the slices these rectangular or just the bounding boxes of where the tumor is located at. Now, we did encounter some small inconveniences because we are not radiologists, so we realized that only using CT might not be enough in all cases. Thus, out of the 355 patients, we chose to work on the ones who also undergone PET scans, so 2 image acquisition methods were used, and we just chose them. I can show you how we did that. We did use the clinical data, so I have here the Excel sheet with all the patients, out of which, so 355, out of which this is the whole list. We chose the ones who undergone both CT and PET. This led us to only having 2 types of cancer in the end. Amin and Bas worked on the ones who were diagnosed with adenocarcinoma, and me and Razvan to the patients with G, so squamous carcinoma. We were able to collect these, if this would load, by selecting as an image modality all and then just CT and PET. We looked at all the subjects, decided on whether or not we want to work with one or multiple studies. This was really different based on the gender as well. We initially wanted to work on 2 males and 2 females, so for each type of cancer, 1 male and 1 female. But unfortunately, for my cancer that I chose, for the type of cancer that I chose, there was not enough data for females. So we decided in the end to only work on males. And how we actually chose which patients to use out of the 355, we all wanted to have multiple T stages, so to see in time how the tumor grows, so that we can have nicer visualizations of the tumor and try to get a bigger or better understanding of these types of tumors. So for instance, Amin had the patients in blue, the rows in blue, and for T stages he had 2B, 1C, 4, 2A, and 3. So all different T stages, we all try to have different ones. For G it wasn't possible because the data set didn't have enough data from that point of view, but we still try to not have more than 2 patients who had the same T stage. Alright, this is not loading, but the point was that maybe I can reload the page. Hopefully it will work now. Yeah, okay, good. So we want both image modalities, and for G we have to go a bit more. I'm gonna work on mine just for the purpose of this video. Okay, yeah. So I personally had the ones in purple, which are G36, 38. Yes. 42, 44, and 46. These are my 5 patients, and in total the files have 4 gigabytes. So after I download them, I can just use the manifest, open with NBI Data Retriever, the free app. This differs a bit on Windows, but it's basically the same approach of loading the data. I'm gonna create a folder to save these. Okay, and then I chose a directory. I'm just gonna wait for these to download. It's gonna take some time, and then I'll be right back. Great, the download is complete. Now I have access to my DICOM files, and what my task was for the project was to focus on the metadata and also to determine whether or not there is a correlation between the stage of the tumor and all the other characteristics of the patient. For instance, their age, if they smoked before. I will get back to that in a second. I did a Python code for it, but quickly regarding the metadata, according to my list of patients, we have their unique worldwide identifier, the modality used, whether they were fully scanned, their whole body was scanned, or just from head to the thoracic cavity, how many slices we have, the storage it takes when I downloaded it. So it's really convenient to have everything at one place. Okay, now if I just go to my code, I try to make a simulation based on, well, initially I wanted to work only on my 5 patients, but to get a better understanding I also included response. So we have 10 for the correlation metrics. These are all the in my dictionary I added all the relevant info about each patient, their age, their weight, T stage, normal stage, metastasis stage. This one I could have omitted because there was only one who had it, but regarding the cancer cells, and also this one, the smoking history, which we really wanted. I have here the data frame and plotted the data frame and plotted on each axis the values. It's easier to just look at the correlation heat map. I had to convert some of the strings to numerical values, and also for the histopathological rating, because I didn't have data, I just used unknown, but in the end I got what I wanted, what I was actually interested in about, and surprisingly it wasn't half bad, because with only 10 patients I thought it might not be necessarily enough to have a conclusion, and that is true up to a point. Like there is a strong negative correlation between the weight and the tumor stage of the patient, which could imply that as the tumor grows bigger, so in time the patient might lose weight. But if we were to look at weight and M stage, so in time, based on this, in time the patient would lose weight, the tumor would grow, but then it wouldn't move to other regions of the body. So that was interesting in my opinion. Also regarding smoking history, it didn't have, surprisingly to me, I was expecting that if you would have been smoking for your whole life, or some part of it, the tumor might also be caused by it, which doesn't necessarily happen. There could be a moderate negative correlation for the M stage, so it did decrease, or well, actually increase based on our previous point, the value. But yeah, if we were to do this project in the future again, I would definitely use more patients. I would like to also focus on the gender more, also the age, because the range is not necessarily that different. We only had one patient who was 90, but the others were between 58 and 60. So yeah, we could have chosen that a bit better. But then again, the main reasoning behind our selection was the stage of the tumor, so that we can look at the locations and understand if maybe they have any sort of correlation with the characteristics of the cancer, with squamous carcinoma, or if it's just something that happens in particular for patients who have lung cancer.